Well, welcome to the RNA Collaborative Talks. Today's talks are from the National Cancer Institute RNA Biology Initiative in Maryland. My name is Sandra Wolin, and I head the RNA Biology Initiative. I also wear a second hat as president of the RNA Society. And in this role, I want to encourage all of you to apply for an RNA Society award. The deadline is September 30th, and there are awards for scientists at many career stages. And the details for applying are on the RNA Society website. We have two talks today, and each talk will be followed by Q&A. If you have questions, either during or after the talk, please write your questions in the Q&A. Our first speaker is Natasha Caplen. Natasha is an investigator in the genetics branch of the National Cancer Institute. She received her PhD from the University of London and carried out postdoctoral training, first at Imperial College London, and then at the National Human Genome Research Institute at the NIH. Afterwards, she joined the National Cancer Institute Center for Cancer Research as a senior scientist. She became an NCI investigator in 2016, and her title is hnRNPs, RNA biosynthesis and disease, insights from a fusion driver cancer.
Thank you, Sandy, and thank you to the RNA Collaborative group. Can I just check that everyone can hear me correctly?
Yes.
Perfect. Thank you very much indeed. And you can see the laser pointer?
Yes.
All right. So again, thank you so much to the RNA Collaborative for giving me the opportunity to present some of our recent work. So for many years, my laboratory has examined aspects of the regulation of gene expression via the RNAi mechanism. And this work included contributions to the development of technologies that exploit this pathway and their use in the investigation of disease states, particularly the identification of cancer dependencies. And I just wanted to point out that we reviewed many of these studies in this recent article. Much of our current work, though, has moved to another aspect of RNA processing, focusing on proteins categorized as hnRNPs, heterogeneous nuclear ribonuclear proteins, RNA binding proteins that regulate many aspects of RNA maturation. And today I'll give an overview of our studies examining the functions of two different hnRNPs, an hnRNPH1, a member of the hnRNP HF protein family, which predominantly functions as a regulator of alternative splicing, and one of the FET proteins, EWSR1, which functions at the nexus of transcription and RNA processing. And we've conducted much of this work using cell line models of the soft tissue and bone tumor Ewing sarcoma. Ewing sarcoma typically arises in children and young adults, and the overall 5-year survival rates for children diagnosed with Ewing sarcoma is about 70%. However, the current treatment regimes are associated with significant risk of secondary cancers or other health issues later in life. Furthermore, for those individuals that fail to respond to upfront treatment or present with metastatic disease, the 5-year survival rate remains poor at around 30%. Thus, there is an urgent need for new means of targeting primary and metastatic Ewing sarcoma. In most cases of Ewing sarcoma, the initiating mutation in this tumor type is a chromosomal translocation that fuses the 5' end of the EWSR1 gene, which, as I said a few moments ago, encodes an hnRNP that functions in transcription and RNA processing, to the 3' end of a member of the ETS family of transcription factors, FLY1 or ERG. The resulting fusion oncogene expresses an aberrant transcription factor that drives tumorigenesis via epigenetic mechanisms, activating genes that promote cell proliferation and repressing regulators of development or cell differentiation. Today, I'll discuss how the study of Ewing sarcoma cell line models is offering us insights into the function of EWSR1, work spearheaded by Sandra, a current postdoc in my group, and performed in collaboration with Dr. Dranka Lansarak, a PI in the CCR's Cancer Innovation Laboratory, and hnRNPH1, studies that has included contributions from several postdoctoral fellows, and it's this work that I'll discuss first. hnRNPH1 is a member of a family of RNA binding proteins that comprises the 5 members shown here. Members of the hnRNPs H and F protein family are highly homologous and contain multiple quasi-RNA recognition motifs and highly repetitive complexity domains. The hnRNPh proteins, including hnRNPh1, preferentially bind G-rich RNA sequences, and studies have linked them to many aspects of RNA maturation, including alternative splicing. Recent studies have associated mutations in this family of proteins to a group of neurodevelopmental syndromes, and these proteins have also been linked to several neurodegenerative disorders. In cancer, studies have reported associations of mutations in hnRNPh1 with mantle cell lymphoma, along with obviously our study of hnRNPh1 function in Ewing sarcoma, which began with its identification as a lead candidate protein in an RNAi screen of EWS-FLI1 transcriptional activity. To understand why this protein emerged as a lead candidate in this screen turned out required consideration of the genetics of Ewing sarcoma. In about 65% of EWS-FLI1-driven tumors, the chromosome 22 breakpoint occurs in intron 7 of EWSR1, while the remaining 35% occur in intron 8. Critical to this story, in the case of those tumors that harbor breakpoints in intron 8, the processing of the EWS-FLI1 transcripts requires exclusion of EWSR1 exon 8 to generate the in-frame mRNA that will express the fusion oncoprotein. The failure to exclude EWSR1 exon 8 results in the presence of a premature stop codon, and thus there is no expression of EWS-FLI1 protein. Importantly, the TC32 cell line in which we formed the RNAi screen harbors an intron 8 breakpoint, leading us to hypothesize that the exclusion of EWSR1 exon 8 requires hnRNPh1. Our follow-up studies show that hnRNPh1 does indeed mediate the exclusion of EWSR1 exon 8 from EWS-FLI1 transcripts. For example, using PCR analysis, we demonstrated that when we silence hnRNPh1 in TC32 cells, we can detect significant retention of EWSR1 exon 8. As predicted, silencing of hnRNPh1 results in decreased expression of the fusion oncoprotein, and critically, this results in reversal of Ewing sarcoma gene expression signature and induces cell death. More recently, by applying long-read sequence analysis, we've confirmed these findings, as seen in these IGV plots of full-length EWS-FLI1 fusion reads in control and hnRNPh1 depleted TC32 cells. Long-read sequencing shows a shift from the exclusion of EWSR1 exon 8 to its retention when we silence hnRNPh1. Interestingly, the number of fusion transcript reads obtained following the silencing of hnRNPh1 is comparable to that observed when we target the fusion transcript directly, consistent with the prediction that the presence of the premature termination codon results in degradation of the misspliced transcripts by nonsense-mediated decay. To date, hnRNPh1 is the only protein identified as selectively required to express any oncogenic EWSR1 fusion transcript. And these findings suggest that the exclusion of EWSR1 exon 8 is potentially a targetable event, at least in 1/3 of Ewing sarcomas. To extend our initial findings, we focused on determining the cis-regulatory sequences that govern the splicing of EWSR1 exon 8 and what these sequences can tell us about RNA structure and hnRNPh1 function. As I stated earlier, hnRNPh proteins, including hnRNPh1, preferentially bind G-rich sequences. And our studies rapidly led us to focus on the G-rich sequences present at the 3' end of EWSR1 exon 8. And the question as to whether these sequences harbor cis-regulatory sequences that can define the inclusion or exclusion of EWSR1 exon 8. And to address this question, we've generated mini-genes, one encompassing EWSR1 intron 7, exon 8, and intron 8, indicated by the E in this slide and the next. And then a second mini-gene that includes EWSR1 intron 7 and exon 8, and a fusion of EWSR1 and FLI1 intronic sequences. Under control conditions in the context of either mini-gene, we observe approximately equal inclusion or exclusion of EWSR1 exon 8, both in the E and in the EF fused mini-gene. But in the absence of hnRNPH1, we observe a shift to the inclusion of this exon. Interestingly, though highly homologous to hnRNPH1, depletion of hnRNPH2 or hnRNPHF had no effect on the splicing of EWSR1 exon 8. Observations consistent with other results we've generated that the exclusion of EWSR1 exon 8 is hnRNPH1 specific. Using our mini-genes, we've next modified the G-rich sequences within the 3' end of EWSR1 exon 8. For example, shown here is the original mini-gene sequence for one of the G-rich sequences in EWSR1 exon 8. And here is where we've made 3 GA substitutions, which results in almost a complete move to inclusion of EWSR1 exon 8. GA substitutions in a second G-rich region present within the last 25 or so nucleotides of the exon also, with one exception, results in inclusion of the exon. And based on these analyses and others, we've thus concluded that the G-rich sequences present within EWSR1 exon 8 are critical regulators of its splicing. This conclusion led us to our next question. Do the G-rich regions within EWSR1 exon 8 form a specific secondary structure? It's been known for several decades that in vitro, in the presence of a cation, such as potassium, G-rich nucleic acids, DNA and RNA can form guanine tetrads that when stacked form a G quadruplex or G4 structure. In vitro-based transcriptome-wide studies have shown that hundreds of G-rich RNA sequences have the potential to form G quadruplexes. And I'm showing you here just one data demonstrating changes in the CD spectra of RNA oligonucleotides corresponding to the 2 G-rich regions in EWSR1 exon 8 when we add potassium that shows indeed these sequences also have the capability to form G4s. However, based on these results and many others, we've predicted that the EWSR1 exon 8 G-rich sequences have the potential to form non-canonical bilayer G4 structures. And one of the questions that we've gone on to ask is then, how would this structure be perturbed if it was bound by hnRNPH1, or would it be perturbed? For this, we developed FRET-based assays in which we could measure whether the oligonucleotide is in a folded state, indicated by a high FRET signal, or an unfolded state, indicated by a low signal. And shown here is just one representative data set demonstrating that in vitro, hnRNPH1's binding can disrupt the G4 folded state. Interestingly, we can recapitulate this transition from a folded to an unfolded state with just a truncated protein that consists just of the QRM1 and QRM2 domains of hnRNPH1. And these studies and many others looking at the biophysical parameters of these interactions are indicated in this study here. But what about in cells? And can we develop evidence that this is a targetable event? For these studies, we began by evaluating the effect of the pan-G quadruplex nucleic acid binder, pyridostatin, PDS. In the representative results shown here, we've observed concentration-dependent alterations in the splicing of EWS-FLI1 and EWS-FLI1 protein expression when we've treated TC32 cells with PDS. This alteration in the expression of EWS-FLI1 results in transcriptional changes consistent with reversing the Ewing sarcoma gene expression profile, including changes in the expression of well-established targets of EWS-FLI1. And these data suggest that quadruplex structures do influence the expression of EWS-FLI1, and we're currently seeking other strategies that make use of this observation or our indication of identification of specific cis-regulatory sequences as approaches for targeting the subset of Ewing sarcomas that are hnRNPH1 dependent. Now, in the last few minutes, I want to turn to our studies focused on another hnRNPH1, EWSR1. Multiple fusion-driven tumors involve genetic rearrangements that involve members of the FET protein of FUS, EWSR1, and TAF15. The pathogenesis of several neurodegenerative disorders are also involved these proteins. The FET proteins contain RNA and DNA binding domains, as well as multiple low complexity domains. Previous studies have highlighted the FET protein's functions in regulating basal transcription, particularly its regulation of the phosphorylation of RNA pol II, and their involvement in the response to DNA damage. However, many of these studies have, often by necessity, used biochemical assays or the expression of non-physiological levels of the FET proteins, and thus we lack detailed assessment of their functions in cells at endogenous levels of expression. To begin to address this deficit, we've used Ewing sarcoma cell lines to generate cell-based systems expressing only endogenously tagged EWSR1 protein. In brief, we used a knock-in CRISPR-Cas9 strategy to modify two Ewing sarcoma cell lines, A673 and TC32, to express the M neon green reporter protein fused to the M terminus of endogenous EWSR1. Following validation of selected single-cell modified clones, we conducted systematic image-based analysis using standard confocal microscopy, 3D structured illumination microscopy, or SIM, 3D SIM, and stimulated emission depletion STED microscopy. I'm happy to discuss further technical details after this presentation, but for now I want to point out that 3D SIM instead enables us to image fluorochrome stains such as DAPI, the M neon green fused EWSR1 protein, and proteins detected using antibodies at a resolution of up to 120 nanometers. STED microscopy also enables us to resolve antibody-based detection of specific proteins at a higher resolution, which in the context of this presentation, we've examined at a resolution of about 60 nanometers. As further background, we've obtained the images shown today using procedures that minimize loss of nuclear volume, and we've generated the 3D reconstructions using at least 20 Z-planes across the central region of a nucleus. The results I will present today will assess the overall organization of EWSR1 and its cellular localization relative to DNA and chromatin markers, as well as markers of transcriptional regulation and nascent RNA. Please note we've conducted all the studies presented here in both A673 and TC32 cell lines with comparable results. However, today I will predominantly present representative images of modified TC32 cells. Shown here are representative images of modified M neon green EWSR1 Ewing sarcoma cells using super resolution confocal microscopy, which shows that the modified protein retains the nuclear plasmic localization of endogenous EWSR1. We also observe that M neon green EWSR1 is excluded from the nucleus. We next assess the kinetics of endogenous M neon green EWSR1 by fluorescence recovery post-photo bleaching, or FRAP. FRAP analysis demonstrated that EWSR1 is present in both a faster recovery fractions, as indicated by T half-lives of less than 10 seconds, and in slower recovering fractions, as indicated by T half values of over 50 seconds. These results are consistent with endogenous EWSR1 protein existing in two kinetic states within cells, one in which the protein is undergoing long-term interactions and one consisting of rapid dynamic interactions. Based on these findings, we hypothesize that these two kinetic states reflect EWSR1's varied binding of different macromolecules, for example, nucleic acids and proteins. Shown here is a representative nucleus with the M neon green EWSR1 signal pseudo-colored in gray, including a line plot of its intensities across the indicated insert. And we've observed that endogenous EWSR1 exists in two visual modalities, one which we've termed distributed due to its diffuse signal throughout the nuclear plasm, and the other as foci. Both visual modalities exist throughout the volume of the nucleus, as seen in this video. To systematically analyze foci across samples and cell lines, we applied a threshold of at least 3 times above background and a size restriction of above 20 nanometers. And applying these thresholds, we've quantified about 25 foci per central Z-plane of a nucleus that vary in size from about 200 to 1,000 nanometers. To assess EWSR1's localization relative to DNA, we first used DAPI staining and histone H3 to define condensed regions of DNA as marked by excess DAPI staining, and euchromatin as defined by less intense DNA DAPI staining and enhanced detection of histone H3. Analysis of the DAPI and M-neon green EWSR1 signals revealed exclusion of M-neon green EWSR1 from condensed regions of DNA, while analysis of the histone H3 and M-neon green EWSR1 signals indicate that these proteins are present in the same nucleoplasmic space, specifically euchromatin. We next assessed EWSR1's localization relative to the transcriptional regulators BRD4 and marker of mediator complex MED1. And in both cases, we observed that EWSR1 is present in the same nucleoplasmic space as these proteins, with EWSR1 foci adjacent to these transcriptional regulators. In contrast, and inconsistent with biochemical studies, visualization of EWSR1 with phosphorylated RNA pol II in our reported cell lines indicates significant co-localization of the M-neon green EWSR1 foci and RNA polymerase II in states associated with transcriptional initiation and elongation. Finally, we visualized EWSR1's spatial organization relative to nascent RNA using EU incorporation combined with click-it chemistry. The video across these steps and the representative images show that M-neon green and RNA exhibit high degree of overlapping signal throughout the nucleoplasm, but not the nucleolus. The representative line plot shown here indicate that both visual modalities of M-neon green EWSR1 distributed and foci associate closely with nascent RNA. In conclusion, we've now benchmarked EWSR1's overall cellular organization, showing that it's present in two dynamic states and two visual modalities, foci and distributed. And EWSR1 in both states associates with RNA. EWSR1 foci associate with euchromatin and are present adjacent to BRD4 and MED1 and overlap with a substantial subset of phosphorylated RNA polymerase II. And these findings are consistent with previous biochemical studies, but also represent the first high-resolution analysis of any endogenous FET protein. We're now using these model systems to interrogate EWSR1's interactions with other macromolecules and comparing its distribution with that of other hnRNPs and its response to external stimuli. And we present some of our initial studies of FUS's distribution compared to that of EWSR1 and EWSR1's response to the dual CDC7 CDK9 inhibitor in the manuscript that's available at bioRxives. Our work at investigating hnRNPH1's function is also continuing, focusing on further analysis of its interactions with RNA and with other proteins, and examination of different strategies for selectively inhibiting the exclusion of EWSR1 exon 8. And we're also interested in determining if hnRNPH1 mediated exclusion of EWSR1 exon 8 is an adaptation of the expression of one or more transcript variants expressed by EWSR1 or FLY1 or a neomorphic event. Finally, I want to again acknowledge current and previous members of my team that have conducted the studies presented today. And I really want to highlight the expertise and assistance of our colleagues, particularly Dr. Janka Lazarek, who helped with the high-resolution microscopy, and Jay Slethkoff with the work on G quadruplexes, and also other people that we've collaborated with, and the NIH and NCI cores listed with that. And with that, I'm happy to take questions.
So we have a question in the chat, and please write your other questions in the chat. So the first question is, how specific is PDS in controlling the exon 8 splicing of EWSR1? So
unfortunately, PDS is a pan G4 binder. It can actually bind both RNA and DNA. We see that this is when we add PDS to cells and then look at its splicing, we see that this is a very, very early event, and it's a very dominant event. And in fact, in the context of other splicing events, but it unfortunately is very non-specific. And is one reason why we're now trying other strategies to selectively target this event, for example, using switch splicing oligonucleotides.
Any other questions?
We covered a lot of ground.
Covered a lot of ground, yeah. Do you want to say, yeah, so you just talked about your attempts to, what you're doing going forward to try and get specific inhibition. Do you want to expound a little bit on that, or?
Yes, so we've tried a number of different strategies to try and target this particular event selectively. So obviously, there are other molecules that are being developed that bind to G4s or thought to interfere with their form, and we've tested some of those, but as yet none of those has been as effective as a protostatin. We've also considered and have done some pilot experiments looking at trying to use decoys. So you're taking a folded RNA and see, or that would that's mimicking that, and seeing if that would bind to hnRNPH1. We had some data that was suggesting that that may work, but again, it lacks specificity that we were looking for. So at the moment, what we're pursuing predominantly is trying to use oligonucleotide-based interactions with the cis regulatory sequences that we've identified as binding to hnRNPH1.
So if anyone has additional questions, they can continue to write them in the chat. And if Natasha sees them, she'll answer them.
Of course. Thank you very much.
Yes, thank you. So our second speaker is Sarah Dremel. Sarah obtained her PhD at the University of Pittsburgh and is now a postdoctoral fellow in the laboratory of Joe Ziegelbauer in the HIV and AIDS malignancy branch of the Center for Cancer Research, National Cancer Institute. And her title is Consequences of Circular RNA Escape During Herpesvirus Host Shutoff.
Thank you so much for letting me speak. I'm really excited to talk about some of my recent work in herpes viruses. So first, considering this is an RNA biology audience, I wanted to just give a brief overview of the pathogens I study. So there are 9 species of herpes viruses which infect human. Phylogenetically, they're separated as alpha, beta, and gamma. And today I'll be discussing some of these different species, including herpes simplex virus type 1, which commonly causes oral and genital lesions, cytomegalovirus, which is the leading cause of congenital defects. And then, of course, I'm from the NCI, so I do study cancer, 2 different oncogenic viruses, Epstein-Barr virus and Kaposi sarcoma herpes virus. So we lack vaccines for all but one of these viruses. There is no therapeutic agent that can target the latent reservoir, and particularly for KSHV-driven cancers, we really need better biomarkers for their diagnosis. Human herpes viruses are large double-stranded DNA genomes, so they encode upwards of 100 different genes, and these include both protein coding and non-coding RNA. They have a biphasic life cycle. So once you have a herpes virus, it will persist with you for the rest of your life. That's why they're so prevalent. And that's when the virus stays latent. But it can also undergo lytic reactivation, which is the replicative phase of infection. And at least for today's talk, I'll be focusing on that lytic and replicative phase, okay? Herpes viruses are nuclear replicating, so they undergo most of their steps inside the nucleus, and they rely on the host transcription machinery for creation of their transcripts. Okay. So the aspect of herpes viruses that I study actually is something kind of interesting called circular RNAs. And they're an alternative splicing product, which is formed by a by a 5' to 3' covalent linkage called a backsplice junction. They're usually thought to function in gene expression by serving as microRNA sponges, RNA binding protein modulators, and transcriptional enhancers. However, they are not strictly non-coding and actually can serve as translational templates by CAP independent translation. So I just labeled this slide fun facts, but it's kind of things to get you interested in this type of transcripts. So circular RNAs have extended half-lives, and that's because they don't have ends and they're resistant to exonucleases. So their stability is at least 2-fold longer than linear counterparts. They're generally considered non-immunogenic. They can be found in extracellular vesicles and particles, and actually up to 50% of extracellular RNA detected in plasma is circular. They are widely expressed throughout our body with tissue-specific profiles, as well as disease-specific profiles shown in cancer, neurodegenerative diseases, cardiovascular, and diabetes. And that's kind of the start of what I'm going to be talking to you about, is expression profiling in the context of herpes virus infection. So here I'm just showing you a big overview of all of the different models that I assessed by RNA sequencing. For the purpose of today's talks, what I really just want to orient you to is that I had virus models that spanned the alpha, beta, and gamma herpes viruses. These included both primary cell culture as well as animal models of infection. And again, they also spanned both the lytic and latent models of the life cycle. So how do we profile circular RNAs? A lot of you have probably never even heard of them, and that's because in a standard Illumina short read sequencing experiment, they actually get thrown out because you either do poly A selection, and these things don't have tails, or you throw out the reads in most pipeline tools because they're chimeric splice junctions. So we specifically look for these chimeric splice junctions using a custom pipeline, and that's how we analyze both forward splicing as well as back splicing events. So that's what we did. And here I'm just showing you Venn diagrams for upregulated human circRNAs in 3 different models of herpes virus infection. And this is specifically looking at lytic infection. So here you can see that we found a number of different upregulated circular RNAs in an alpha, a beta, and a gamma herpes virus. And across these different, very different viruses, different cell types, we actually did find 4 circular RNAs in common between these different models. So the next question, these are an alternative splicing product. So are there collinear gene products similarly upregulated? Now here I'm showing you those differentially upregulated circRNAs in a heat map form. So anything in purple means it's upregulated with infection. Anything in orange means it's downregulated with infection. So again, I'm limiting this to showing you the upregulated circRNAs. So you can see in purple, they're upregulated with infection. Now it's really quite obvious for HSV1, all of the collinear mRNA products are orange, which means they're drastically downregulated by infection. And you can see a similar trend to a lesser degree for the other viruses. So another way that we can think about and quantify this is something called a CIRC score. And that essentially measures which isoform is more prevalent. So anything that's more blue means the circular RNA is more prominent than the collinear mRNA, and yellow would be even more so. So as you can see, as we progress between uninfected, 12 hours post-infection, and really late times post-infection, there's this shift in favor of the circular RNA isoform. And again, you can see this shift as well for the other viruses here. So here I showed you that there's disparate expression changes for circRNA and their collinear mRNA. But what does this look for the entire population? I was just showing you a subset. Okay. So here I'm plotting RNA sequencing reads for all protein coding transcripts, and I'm plotting it as a log 2-fold change of infected over uninfected. So you see this nice normal distribution, but you also see that there's a shift downwards with infection. So there's a pretty drastic decrease in pretty much all mRNA with infection with a median log 2-fold change of -4. Okay. How does this look for circular RNA species? Now here, when I plot circRNA in the same method, you can actually see that they're largely unaffected with actually a subpopulation that's drastically upregulated with infection. Now circRNA, again, they are generally considered to be non-coding RNA. So maybe this just shows a difference between protein coding and non-coding RNA. So I plotted the same kind of thing for lncRNA, long non-coding RNA. And here you can see that they look more similar to the mRNA distribution shifts, where there's a median log 2-fold change of -4 for lncRNA between uninfected and infected samples. So this analysis, this is for the alpha herpes virus HSV1. What about a different herpes virus? Do we see something similar? So here I'm showing you the same analysis for the oncogenic virus KSHV. Again, you can see for both mRNA and lncRNA that there's this shift downwards in the population with infection. Now, by comparison, again, circular RNAs are largely unaffected. So they're escaping whatever's going on here. Okay. I have one more virus to show you. So this is actually a mouse gamma herpes virus, MHV68. Again, albeit maybe to a lesser extent, you can see this shift in the population for mRNA and lncRNA with infection, where they're being downregulated, as a global population. By comparison, again, circular RNAs are largely unaffected by this. Okay. So circRNAs, as I said, they're an alternative splicing product. And what we can do then is actually analyze reads that map to forward splicing events, so mRNA, versus the circular RNA reads. And we can do that for a given gene. So for a gene, same gene should have the same sequence largely between the circRNA and the mRNA. How exactly is that being impacted by infection? And here again, I'm plotting as a log two-fold change. And it's kind of segregated by quadrants. You can look at within a given gene, how is the circRNA versus the mRNA impacted by infection? So here I'm showing you data from my 3 different viruses. And as you can see, again, this is really notable for HSV1. The data is falling in this bottom right quadrant, which means for each of the genes that I analyzed, circRNA is being upregulated by infection, but the collinear, very similar sequence in the same gene, mRNA, is being downregulated. For KSHV and MHV68, which are gamma herpes viruses, I see a similar trend where in every instance, the mRNA is more downregulated by infection than the circRNA. So why is this? Why do we observe disparate expression changes between circRNA and their collinear products? So I'm sure everyone in this audience is familiar with this kind of concept of mRNA decay. We can proceed through 2 different options. One, which can be mediated by an endonuclease, and then the final step occurs through an exonuclease. Well, as you can imagine, circular RNAs don't have ends, so this top option is not available for them. However, they're still susceptible to endonucleases, and then if they are cleaved by an endonuclease, they can then be targeted by exonucleases for degradation. And this has been shown to be true for the human endonucleases, RNase L and RNase P, that they can target circRNAs and lead to degradation. However, what maybe the audience doesn't know is that herpes viruses actually encode their own endonucleases. They're encoded by the alpha and gamma herpes viruses. There's also endonuclease encoded by influenza as well as coronaviruses. And I just want to highlight that all viruses known to drive widespread mRNA degradation do so by causing internal cleavages or by directly removing the mRNA 5' cap structure. So there are disparate mechanisms, but viruses have their own endonucleases. So my thought was perhaps circular RNAs are resistant to these endonucleases. Okay, so to test that, I transfected cells with viral endonucleases and did qPCR measuring the levels of circRNA and their collinear mRNA. So everything here is endonuclease versus a GFP transfection control. And in blue would be circular RNAs, in yellow is mRNAs, and one would be baseline expression. As you can see in every single instance across the board, in blue, the circular RNA is more resistant to endonuclease expression than their collinear mRNA. And this is quite drastic, especially for VHS encoded by HSV1. So kind of quick recap of this part is some of these really disparate expression changes for circular RNAs during herpes infection seem to be due to this difference in decay. And that is largely attributed, I think, to the difference in susceptibility to the viral endonucleases. Now, I want to switch gears. I talked about this and how that might be contributing to gene expression. But what about this step? What about synthesis? So what common pathways across the different viruses and cell types might be driving circRNA expression? I found a number of circular RNAs that were upregulated in my different models, different viruses. What might be in common? What might be driving that? So here I'm showing you just normal RNA-seq pathway analysis for my 3 models of lytic herpes virus infection. And what I want you to see is that among these most significantly upregulated pathways, we're seeing a lot of different things that speak to cellular immunity, okay? So across the board, you're seeing an upregulation of cellular immunity. Not super surprising, these are infected samples, right? And if you look at predicted upstream regulators, both type 1 and type 2 interferon are highly predicted to be, upstream drivers of these gene expression changes. So is it possible that some of our circular RNAs are actually part of this response? Might they also be interferon stimulated? So to test this, I took cell lines that are what we usually will do our infection models in. So these span both fibroblasts lymphatic endothelial cells, as well as B cells, and I treated them with interferon beta or gamma. Again, I did RNA sequencing and chimeric junction analysis to look at circular RNA levels. So these are volcano plots, and any of the dots in green are circular RNAs that were upregulated by interferon stimulation. And I've just highlighted a couple of these, and I'll come back to them later. One of these, Circ-EPSTI1, was actually found to be upregulated in both type 1 and 2 interferon treatment, and in all 3 of these very different cell types. There's also this circular RNA called Circ-REL1 here that we found to be upregulated by interferon treatments. So another way of looking at this is plotting the levels of circular RNA change against mRNA chains within a gene, with the thought being that if these are being driven, similar to what we think of interferon stimulated genes, we have these transcription factors, they bind to the promoter, they drive expression, that we would see similar changes in expression for the circ and the linear form. So here, I'm showing you that. So anything on a diagonal has similar levels of upregulation between an mRNA and a circ RNA. And we find that there are a number of these that have that trend. These include things that I think some of us have heard of in immunity, like SP100, B2M. EPSTI1 is actually a interferon stimulated gene as well, WARS1. So there are definitely some of these that seem to be following that classical, probably being controlled at the level of transcription mechanism, but there are others where the circ RNA is upregulated, but its collinear mRNA is not. So this might speak to secondary mechanisms for upregulation, things like RBPs, changing splicing, or decay possibly as well. So are interferon stimulated circular RNAs antiviral? So here, I'm just showing you a Venn diagram looking at all of the different circular RNAs we found to be upregulated in our different infection models, and then all of the different circular RNAs that we found to be upregulated by interferon treatment. And there's actually an overlap here where about half of those interferon stimulated circular RNAs are also upregulated by herpes infection. And among these is one that I kind of referenced to earlier called Circ-REL1. And our lab has actually worked on this circular RNA in the past. My colleague Takanobu Tagawa published a paper just this last year discussing how Circ-REL1 is anti-lytic within the context of KSHV infection. So I found that Circ-REL1 is interferon stimulated. We know that it's anti-lytic for KSHV, but I also found that Circ-REL1 is upregulated in additional models of herpesvirus infection, including HSV1, CMV, and EBV. So could it perhaps have a pan antiviral role? So to assess this, what I did is I did both gain and loss of function assessing Circ-REL1. So I used lentiviral transduction to compare Circ-REL1 overexpression to a Circ-GFP control. Cells were infected at a high MOI for 12 hours, and then I looked at the amount of new virus made from the model. Alternatively, I did siRNA transfection, where the siRNA can specifically target the backsplice junction or unique sequence of the circ RNA to cause depletion. Again, a high MOI infection, and looking at viral yields. So here I'm showing you qPCR data, and in our gain of function model, when we measure Circ-REL1 expression, we see that we have, in fact, a gain of function. We have overexpression of Circ-REL1. By comparison, when we do siRNA transfection, we now have a significant drop in Circ-REL1 levels. When we measure viral yield, so again, this is looking at the amount of virus made at the end stage of infection, we can see that with Circ-REL1 overexpression, we have a drop in viral yields. Whereas with Circ-REL1 depletion, now we have a corresponding increase in viral yields. So both gain and loss of function models agree, and it seems like Circ-REL1 is antiviral in the context of lytic HSV1 infection. And then with our prior findings regarding KSHV, it seems that this molecule might be pan-herpesviral. So it's able to act on multiple viruses in multiple cell types and restrict lytic infection. So that's one example that we were able to look into. But to see what maybe some of these other things are doing, we did in silico predictions. So circRNAs have a well-published role that they will serve as microRNA sponges. And in that way, they control mRNA gene expression by restricting microRNA decay. So what we can do is look for circRNA microRNA binding sites, and then subsequently predict the messenger RNA that might be impacted by this interaction. So here I'm showing you some of those in silico predictions. And this is for the 4 different circular RNAs that were commonly upregulated across alpha, beta, and gamma herpes viruses. So when I do this kind of analysis, then I can look at the messenger RNA and see what kinds of downstream pathways they might be enriched in. And as you can see here, there's a lot of different pathways. These all seem to be related to adaptive immunity, peptide presentation. So there's this enrichment for circular RNAs being upregulated during infection that have at least an in silico predicted role in cellular immunity. So this kind of brings me to my overall model, where herpes viruses, and in part because it's likely part of an intrinsic immune response, stimulate circRNA and mRNA expression. However, mRNAs are susceptible to both viral and host endoribonucleases. In particular, this is likely mediated by the viral endonucleases, whereas the circRNAs are resistant to viral endonucleases and escape that decay. These can in turn go back and actually restrict the virus. Now, this is, we showed this for one circRNA. There's a number that we found now in our profiling studies to be upregulated during infection or stimulated by interferon. And that's very much something we're super interested into looking at more. We're also very interested in looking and thinking about the ways that the circRNAs may be upregulated outside of just the transcript itself being turned on. So those are kind of places that we're going next with this. And if you want to learn more, the paper's up on bioRxiv. So with that, I just really quickly like to thank everyone in my lab, particularly another postdoc, Takanobu Tagawa, who I worked with on this project, our collaborators that helped us with some of the different models of virus infection, as well as the chimeric splice junction analysis, and everyone who's given us reagents to do this study. And with that, I'll take questions.
So thanks, Sarah. That was great. So please put your questions in the chat. So the first question is, have you investigated virus synthesized circRNAs?
Sorry, one sec. There we go. Okay. Can you repeat the question? I'm sorry.
So have you investigated virus synthesized circRNAs?
Yeah, great question. That's actually what I focus on in the lab primarily, is virus synthesized circular RNAs. So viruses very much do make their own circular RNAs. It's been shown for DNA and RNA viruses. Herpes viruses make a dozen of different circular RNAs, and they're specific to either the lytic or latent mode of infection. At least for the oncogenic herpes viruses, it seems like they might play a role in promoting cell growth, something obviously of concern for a cancer-causing pathogen. So yeah, definitely viruses make their own circular RNAs, at least for some of them that have been shown.
So next question is from Lila Lackey. If you look at the predicted mRNA targets from your possible microRNA sponges, do you see any expression differences in your RNA-seq data?
Yeah, it's a great question. I've kind of played with that analysis. The problem, as maybe you can guess, based on some of the plots I showed you about widespread mRNA changes, is that there are a lot of different ways that herpes viruses shut down host transcription. So it happens at the actual transcriptional level, post-transcriptional level, and as well at RNA decay and export. So I haven't done this because I think it will just be too messy. Honestly, especially during HSV-1 infection, it causes just really drastic changes very fast, where in a period of maybe 8 hours, you'll see viral host transcripts, going from 100% to 50% or less of the total RNA in a cell.
So we have a question from Vladimir Machursek. Hi, Sarah. Nice talk. Did you try to correlate the subcellular localization of mRNA/circRNAs and their sensitivity to degradation? Most of virus-induced RNA degradation occurs in the cytoplasm. Can nuclear accumulation of some circRNAs prevent their degradation by virus-encoded endonucleases?
Yeah, that's a great question. It's generally thought that a bulk of host circRNAs are cytoplasmic in their localization. So theoretically, based on that, they should be susceptible to the viral endonucleases. They'd be in the same compartment. However, who knows? During infection, it's very possible that the subcellular localization of these circRNAs changes. At least for HSV1, I think the mechanism of not being targeted is pretty clear. And that's because the HSV1 endonuclease specifically targets translation competent transcripts. So it's actually being brought to its substrate through association with the translation machinery. So that's kind of a cut and dry reason why circRNAs are probably escaping. For the gamma herpesviruses, they're actually targeted through a sequence-specific motif. So in that way, circRNAs absolutely should still be susceptible. Some kind of thoughts we've had for why they wouldn't be targeted are absolutely, it could be localization. CircRNAs are also shown to really be highly occupied by RNA binding proteins, which would occlude substrate recognition. And they also are shown to be pretty highly structured, again, something that would prevent targeting by an endonuclease.
So I'm fascinated by circRel1. You didn't have that in your list of, was that one of the ones you think is a sponge, or do you know more about how it functions?
Yeah, so that is actually primarily work done by another postdoc in our lab, Takanobu Tagawa, and he's done some really fantastic mechanistic work on it, where he's looked at both RNAs that circRel1 interacts with, as well as proteins. And it seems like, and this is probably the reason it might be just pan-viral in its effect, is that it likely targets somewhere in the mTOR pathway, which we understand, obviously, that's a huge pathway. It's very much a target of herpes viruses and infection. And likely that might be a way that it could be antiviral to multiple viruses, multiple cell types. So it's also possible it might extend past herpes viruses. We don't know yet, but that could be really interesting.
Great. So if there aren't any further questions, let's thank both of our speakers for a great talk. And please remember to apply for an RNA Society award. Okay.